To end the neural control chapter, we're going to talk about different types of sensory receptors. This leads to our outcomes, and so we're going to break things up into three different categories: motor neurons, sensory receptors, and muscle receptors. Now, there are three types of motor neurons. First, we have alpha motor neurons, which are relatively large motor neurons which innervate skeletal muscle and cause the muscle contractions that generate movement. Specifically, they innervate extrafusal muscle fibers, which are standard muscle fibers. These are the ones that produce force that allow you to move. Next, we have beta motor neurons. These also innervate extrafusal fibers, okay, so just think like muscle fibers, as well as intrafusal fibers, and they innervate things like muscle spindles that we'll talk about here in just a minute. Now, as opposed to alpha motor neurons, which actually cause contraction, beta motor neurons are more of a sensory type neuron. And so they are innervated by both motor and sensory fibers. Okay, and then last we have gamma motor neurons, which respond to stretch receptors in the skeletal muscle. And these are those muscle spindles that I told you about a second ago, and ones that we're going to review here in a few slides. Now, although gamma motor neurons are actually referred to as motor neurons, they don't really cause any motor function. Instead, they're thought to be activated at the same time as alpha motor neurons and really help fine-tune the muscle contraction that's going on with the muscle. Now then, there are other types of sensory receptors such as those listed here. So we've got photoreceptors, chemoreceptors, mechanoreceptors, thermoreceptors, and nociceptors. Photoreceptors are activated by photons or light waves and are used in vision. Chemoreceptors are activated by chemicals, and there is a wide range of functions that these serve. Okay, so for example, monitoring how much oxygen's in your body so you know what your heart, or so we can affect heart rate or cardiac output or respiratory rate, things like that. Mechanoreceptors are activated by a change in their shape, okay, or some sort of physical deformity, or vibration, that sort of thing, and these are generally found in your skin. This is what allows you to touch and feel stuff. You've got thermoreceptors, which are generally in your skin, but you also have thermoreceptors internally as well. And these are sensitive to changes in temperature. And then finally, we have nociceptors, which are activated by painful stimuli, and we have those everywhere. Finally, there are four types of muscle receptors. We've got muscle spindles, Golgi tendon organs, Pacinian corpuscles, and Ruffina endines. Now, with each of these, there's two things that you want to know, and I believe we discussed this in lab as well. It's what kind of stimulus does it require? Is it a large stimulus, high stimulus, or low stimulus? And then does it cause high activation or low activation of a muscle? Or basically, are we activating or inactivating the muscle? So muscle spindles are a type of interfusal fiber. They sense muscle length and the rate of change in muscle length. When a spindle is stretched, it sends signals via afferent neurons, which ultimately cause muscle contraction. Now, gamma motor neurons keep the spindle taut, and during normal contraction, we don't want the spindle to prevent lengthening of a muscle, so gamma neurons inhibit spindles through a process called autogenic inhibition. Okay, and so these cause muscle activation. In other words, they contract the muscle, and it takes a relatively high amount of stimulus. Next, we have Golgi tendon organs, and these are unsurprisingly found in muscle tendons. They sense muscle tension and the rate of change in that tension. Now, when a Golgi tendon organ or a GTO is stretched out, it inactivates the muscle to prevent injury. This is why you immediately drop something that is extremely heavy. So just say someone gives you a box, and that box is just really, really heavy. It's full of lead plates or whatever. More often than not, you're just going to immediately drop that, and this prevents injury. Now, in untrained individuals, these GTOs are activated relatively early to prevent injury. However, as a person trains, the GTOs are basically, they aren't activated as quickly. They're activated later. And this actually makes it more likely for someone that's trained to get injured. And elite athletes suffer more catastrophic injuries than those of us that are untrained. Okay. Then we've got Pacinian corpuscles, which are a type of mechanoreceptor that responds to changes in pressure. They're activated by relatively small amounts of stimuli, but they only cause small amounts of activation. Okay, so they cause muscle contraction. Now, an example of activation of these Pacinian corpuscles is when you go on a road trip. When you get out of the car after a long drive, you are stiff because of the muscle activation that has been caused by Pacinian corpuscles. And then finally, you have Ruffini endings. These receptors are activated by low-frequency stimulation that occurs when the skin is stretched out. Just like Pacinian corpuscles, they cause small changes. However, instead of activating the muscle, they cause muscle inactivation. Deep tissue massage activates Ruffini endings, which ultimately leads to muscle relaxation. Okay, that's essentially what they're trying to do. Deep tissue massage, they're trying to activate these Ruffini endings. So are you able to answer these three questions? Can you compare and contrast the three types of motor neurons? Can you compare and contrast the different sensory receptors? And can you compare and contrast the four types of muscle receptors?